Yeah, Dakota is sick. So those of you who aren't with us, we miss you. And here's the lecture quiz that we had today. We're going to go over it really quickly and see how you did. Okay. So what do the letters HPV stand for? Human papillomavirus. And what does this cause? It causes warts, right? And there's over 100 different types. And some of them are very specific to what types of tissues they can attach to. And we talked about that. And how viruses, you know, are respiratory viruses because they're the ones that can attach to the respiratory mucosa itself, right? Where other things are like liver viruses because they can attack the liver. Same thing with these over 100 different HPV viruses. They have different attachments that they can work with. But name the etiology of what is known as the kissing disease or mono. What you put, Jarrett? Because he had asked that right before class. So that's why I wanted him to do that. What is the MMR vaccine protecting against? Measles, mumps, and rubella, which are all viruses. That's exactly right. And what is the DPT vaccine protecting against? Diphtheria, pertussis, and tetanus, which are all three diseases that are caused by bacteria. But you didn't have to put that. You just had to tell me diphtheria, pertussis, and tetanus, right? Distinguish between a direct serology and an indirect serology. Direct is looking for the pathogen. Indirect is looking for the patient's antibody response. Make sure you know that. Okay. What is the genomic makeup of viruses?
Either RNA or DNA.
It's either RNA or DNA. That's exactly right. So it's going to be either an RNA virus or DNA virus. Exactly right. So what is the function of an antibody? To attack specific antigens or pathogens. So they're very specific. They're proteins that are going to be directed, made in response to being exposed to particular antigens, and they will be made to attack those antigens. Any questions about what an antibody is? It's a protein, it's a molecule, right? And it's going to be made in response to being exposed to foreign antigens, and those antibodies attack, destroy, annihilate, take out those specific antigens. Name the #1 viral reason for infant hospitalizations in this country. It's RSV. It is respiratory syncytial virus, right? Rotaviruses cause gastroenteritis. Gastroenteritis. And what is a bacteriophage? It's a virus that infects bacteria, right? True or false? On very rare occasions, the influenza vaccine may cause influenza. Why is that false?
The vaccine is just
So nobody's going to know what you mean when you say that. I call it that, but it cannot cause influenza. It does not. It cannot. It can't. Okay. So so good. That was false. Were these fair questions from the other day? I realized from y'all's study guides that I had plenty of opportunity to write questions. So sometimes I leave here and I think, what did we talk about? Because I didn't even know where the time went. But I realized from your study guides. So thank y'all very much for those study guides. If you haven't done study guides, if you're not really participating, or if you haven't done them recently, that's okay. Just make sure that you get in there and put something in so that you can see your peers, because your peers are doing a fine job of getting this information up there, and you can all benefit from it. So those study guides, I can promise you, are not for me. They're for you, right? They're for you, and it's for you helping your peers as well. All right, so I have a stack of things that I've been stacking up to bring in here, and then guess where they get left? Yeah. Yep, in my office. But then I realized some of them I pull up are a little bit ahead of where we are, what we've gotten to, so I'm gonna have to wait on some of them anyway. But here's one that it's got something in for A&P, my A&P class, but also something for this micro class. And this one, can I go, or did anybody want to go before me? Did anybody hear something in it? I mean, we all know that we're serious, got influenza outbreak, right? Or we're right in the midst of it, aren't we?
I saw an article from before they got it. I saw it on Facebook.
Oh, you saw that that we talked about the other day. Well, everyone should be really cautious here now because we really are in the middle of the influenza season. And so there's just, I just saw something with the numbers of cases right now the CDC put out. I think it's something like 7 million, now of course this is through the country, million cases of active influenza right now. That's a lot, right? So people should be sort of cautious about hand washing and, whatever. But okay, if nobody has anything else, I'll do this. Alcohol-linked disease tops hepatitis C as the #1 reason for liver transplants. And so you've got in your little book with your viruses, that hepatitis C virus. There's a class of viruses that have been designated with letters. So and they just were kind of arbitrarily designated this way, but we call them hepatitis A virus, which is actually very different than hepatitis B virus. Y'all have heard of these, haven't you? And hepatitis C. Well, the thing about hepatitis B and C, and I think I had mentioned this to you guys already, but just make sure you know now because we're introducing it. Hepatitis B and C they're dangerous viruses acutely because you can end up getting inflammation of the liver that when you first get the virus, it can be serious enough, the initial infection, that there's a mortality rate associated with that. But it's really hepatitis B and C, both of them can do this, especially C virus. They can sometimes be subclinical in their presentation. Does anybody know what that would mean, subclinical in their presentation? Below clinical. What does that mean? Yes, that's exactly. So they call it subclinical because the signs and symptoms are kind of under the radar. They're really kind of, it's not even so much that they're asymptomatic without symptoms, but they're not enough that gets them somewhere to be diagnosed early with the disease. So can people have hepatitis C living in their systems for decades? They really can, and this is what we know because sometimes the initial disease is subclinical and it doesn't give you those signs and symptoms that, would have made you go and figure out that you have it. So what they both can do, hepatitis B and C, is actually over time, they can actually cause liver failure. And we can't live without livers, can we? This article is talking about that particular thing. Right now, there are 17,000 Americans on the waiting list for liver transplant. That's a high number, isn't it? Does that seem high? Does anybody know anybody who's had like a liver transplant? Yeah, and they're really sick before they end up getting, at the top of that list, and they're at risk of dying when they're getting pushed up to the top of the list, and usually they're very, very ill. So anyway, they're saying now though that hepatitis C, which had for a long time been the leading cause of people needing a new liver, is now being replaced by alcohol abuse. So anyway, I just thought I'd bring that in because it did talk about hepatitis C, but it's something for us to realize. I think, and I like how they talk about this in this article because it gives us something to think about with medicine and our patients, and also when you are doing histories on patients. They think it's a couple of reasons that the numbers have sort of switched here, and now alcohol is accounting for 36.7% of the people needing livers, where only about 10 years ago it was 24%. But they think it's a couple of reasons that this is happening. One of the reasons, and this is good news, is that people are going to get tested for hepatitis C. And they're finding out if they have hepatitis C because they know that it's a disease that's under the radar. And then they're getting treated. And some people who are getting that treatment are getting cures. So is that saving some people's livers? Yes.
What is subclinical again?
Hepatitis C can be the initial the initial infection with hepatitis C can sometimes go under the radar and you don't know you have it. There are a lot of diseases that do that, so we'll talk about others that can do that too. But the second, and I need y'all to hear this, even though this isn't so much micro-related is that we're finding out people are, through education or knowing, to go get tested for hepatitis C and then getting treated if they end up being positive, because treatment is sometimes curative. But here's the part I need everybody thinking about, because this definitely affects the numbers. And that's that another reason they think these numbers are being pushed up right now is kind of artificial, because people now are more open than they were even a decade ago, especially a couple of decades ago, to admitting alcohol abuse. Before, there was such a stigma associated with it, so people wouldn't really admit it. Now, there are a lot of people who come in, and you're going to know them from your ERs. I can think of one, and he was young because he was in his 40s. One of the last patients I saw die waiting for a liver that was an alcoholic. And sometimes there's enough signs and symptoms of somebody being an alcoholic, they don't need to tell you, so you're going to know. But the fact is our patient history is really important to know. And getting all that information is going to be really, really important to know. Another reason that people weren't reporting it is that even a decade ago, people who said that they used alcohol and they had liver disease, they were never going to be put on a list. They weren't going to be put on a liver transplant list. Why not?
They just ruined the new one.
Because they just ruined the new one. I mean, that's kind of what's the thinking. I didn't say that, but that's Casey said that on the recording. No. But actually, that was the reason, Casey. That is exactly the reason. And in the medical community, you wouldn't give somebody a perfectly good organ that could, if they had behavior that was just going to destroy it.
Is it still like that, or it's not like that?
So it was like that. So now it's changed a little bit. And we understand that, alcoholism is a disease in itself, isn't it? So with what they're doing now is they're letting people know if they have alcoholism, if they have that disease as well. They have lessened some of the stipulations for going on the list. Jenna, your body language is just telling me exactly how you feel about this. Let the rest of the class know.
I think it's stupid.
Okay. Don't hold anything back, okay? So, okay.
Wait,
why do you think it's stupid that they've lessened some of these guidelines?
I mean, I guess in some cases, some people can change, but most of the time, people absolutely will not change, and they're just going to keep screwing up. That's my opinion. I feel agree.
Okay, so what I want you guys to know is that that's why it's taken some time now for us to in the medical community to maybe narrow some of the window of this. Alcoholism, addictions, there's such stigma associated with them because people think about them as they're judged based on having these, like it's something, they're just a bad person, or they just can't get their acts together. I'm not saying this is true. I don't really think this is true. I do think they're diseases. They're diseases. But if somebody were willing to go through treatments for these diseases, because there have been some treatments that are effective for these diseases, and they have a so many month period. Right now it's a 6-month period that they truly are adhering to the treatment, and they truly have proven that they are trying to follow the treatment. They're given a chance. They're put on the list. You see what I'm saying? So I understand, and I hear, does anybody else have how many of you all feel that way? That if somebody has, all right, so thank you for, so it's quite a, Jenny, you're not alone.
I didn't think I was.
Yeah, so you're not alone.
I don't think people are bad necessarily because they have addictions, but I know.
I'm not saying you all thought that. I'm saying that's the way in the past it is seen that way. And I think that's the way it's perceived in the public, you know, that it's that way. And so because of that, people wouldn't even admit that they have the problems.
I just feel like you know if you have an addictive personality or not, and then why would you put if you're setting yourself up for failure?
Well, the reason you do it is because if you're at a place where you're needing a new liver, you do it to live because you're dying.
No, she's talking about taking on alcohol when you know you have addictive personality.
Oh, you mean why would anybody ever drink if you know you have an addictive personality?
Yeah,
Well, I just mean
like anything.
Or drugs or anything? Yeah. Okay. Well, that's a whole nother class, a whole nother topic. So I know it's just really hard, isn't it? Isn't it though? So I think these numbers are affected by both those reasons, that hepatitis C is being more recognized and treated, but also that people who have alcohol and other drug-related problems are more open to admitting and getting treatment for those problems. Yes?
I've heard a lot of people say that, like, alcoholism is genetically passed down.
It is. It is. Addictive behaviors there's genetic component of that. And so this is why I think it's really important too for families that know histories to share histories very early on. Very early on. Usually you don't have to share it very early on with your children because your children can see, you know, that Uncle so-and-so and Aunt so-and-so and mom or dad, you know, they have some, you know, children kind of know, but it should be discussed, you know, it should definitely be discussed. And it is inherent. They do know. They actually know in genetics where it is. Now, there are a lot of alleles that contribute to it, so you end up getting kind of a rainbow kind of effect, but addictive personalities, even with gambling. Yes?
So, I have two questions. The liver can regenerate, correct?
It can.
So the virus prevents the regeneration?
Well, the virus, hepatitis B, C, and A are all doing very kind of different things to it. It's just that it's a cumulative effect of destroying it. So after a time, it will stop regenerating. Yes.
And then how is the weight loss dictated? Like,
Oh, I have no idea. And I think that I know that there's certain things, like they have certain conditions, signs and symptoms and conditions, pre-existing conditions. So if you have A, B, and C of them, but you don't have D and E, that might bump you to here. But if you've had this procedure done, that'll bring you to the top. Do you know what I mean? So it can make you skip over some. Has anybody been involved in that where they've known somebody waiting on the wait list? So sometimes I'm just going to let you know, do you think money plays a role?
Mm-hmm.
Money plays a role. Should it? But also sometimes the money plays a role in kind of a roundabout way, because you'll have a physician that you could afford. And that physician knows that if he could get his patient this procedure done on his patient, it would bump that patient up to the top of the list. Do you get what I'm saying? Mm-hmm. So your physicians play a huge role in trying to jump the list. Now, I'm making it sound like it's such a corrupt thing. I think in anything that's human, you know, we have human biases and whatever, so there's a little bit of corruption in anything human, right? But there are regulating bodies for the transplant guidelines, and they definitely are very much, they're looking at those things. And if they see one particular doctor doing that too often, guess what? He's going to get reprimanded somehow for it, you know. So they do have regulating bodies, the transplant priests. But livers and kidneys now can be living donors. So the good news about it is that you can actually find, you can get living donors. So you don't have to wait until, you know, you've got somebody who's not. Yes, and you said you've known somebody.
Somebody that I know, he was, I think, early 30s, late 30s, and like he had family and everything. And his body produced a protein, and it started attacking his kidneys. And so he went on like the waiting list, and then it got to where like he had no kidney function, so he was on dialysis. And then it started attacking his liver, and then it started attacking his heart. And so he waited for like 9 months, and he got a donor for a heart, kidney, and liver at the same time, the same person. And whenever they went to put the heart in, they said that it might have like a little complication, but they could fix it whenever like they got it in. And so he got the kidney and the liver, but the heart, like whenever they went to put it in, it like messed up, and so it wouldn't work. And so they put him like on like a machine that like circulated his blood.
Blood,
his
sure,
body.
sure.
And so he didn't have a heart for like, I think it was like 16 hours.
Wow.
And
Right
they
now.
found another heart like right around like the area they were in, and it was like a 25-year-old, and he was like older. So I mean, everything went good when they put that in. But
Is this person still with us that
Yeah,
had all these
yeah.
transplants? You know, isn't it amazing? Like people who like I don't understand people who wouldn't do organ donation. That's just such a wonderful thing that people have those chances to come, you know, to do that. And it sounds like that person was really a lucky person.
and he
Yeah.
like moved up really fast on
Well,
the list.
if you're on like that ECMO thing, you're going to be on the top of the list. You're going to get the next one. I mean, anything that's going to be close to your tissue typing, you're getting it because that's so limited how long you can be on it.
Like being a kidney donor or any kind of organ donor, a lot of older people think that if you're going to be a donor, if you're in the hospital right for your life, you could go either way, that the doctors will let you die. So they can take you
Okay. Okay, let me say this, and I don't think I'm naive. Okay, maybe some things, but not really. There's not going to be any doctors that are going to let somebody die so that their organs can be given to someone else. That is not happening. Again, before anything like that could happen, you've got so many built-in safety things like that, but that is not happening. That's misinformation. That's such misinformation that people fear, you know, because it's such an emotional time too. It's like it's horrendous. And so people are fearing that they're going to sacrifice their loved ones so that organs can go to these nameless people. That is not happening. Same way that it's not happening, the late term abortions. Nobody's killing a baby that was delivered. Never has that happened. That is misinformation. Misinformation. Misunderstanding, misinformation, emotions taking hold, amygdalas hijacked. You know, your emotion centers just being hijacked by people who have agendas for something. I'm not sure what. Right? I'm just not sure what. But that is just not happening. Never has happened. If it happened, it's murder. And it has never happened. Not in a medical situation.
I mean, I don't personally think that organ donors, but I know a lot of people do.
I do too, and I'm so glad you brought it up, Casey, because I've heard that so many times. I really have. I've heard that so many times, like maybe they're not going to do everything they could to keep my person alive because they just know that, they just know that the organs can be used somewhere else, and so they're not doing everything in their power. They don't make that decision to have a conversation about organ donation until they have exhausted all possibilities. The person's not making it. You know, they don't even go have that conversation until then.
Jerry, did you have your hand up too?
Yes, this may be a dumb question, but is blood type a factor in?
Blood type is absolutely a factor. Blood type, but then also tissue typing. It has to be tissue typing as well. And size of the organs. So Molly talked about, you said a young person, but an adult. So, you can have an adult who needs organs, but they can't get a child's organs. And vice versa. They have to be able to fit. But that's just whatever. But tissue typing is one of the main things that they have to do, not just blood typing, but also tissue typing. And it's the tissue typing that's the most important to find donors. So, you guys, okay, anybody else have anything they wanted to bring up? We get into the most great things. So we talked about cytopathic effects, and I was so glad that some of you all had put that on your study guides, and you will see that on your next test. So I'll talk about CPEs, these cytopathic effects. Well, we know when we do viral cultures, which we really kind of don't do that often because of the expense, because you know you have to have living cells to have viral cultures. But when we do them, what the virologists are doing is looking for the cytopathic effects to help with their identification and recognition of what virus they might be dealing with. We talked about how viruses can actually lead to mutational changes in cells. And maybe, you know, talking about this, viruses can stay in your system, like hepatitis C that we just talked about, and hepatitis B and herpes simplex. So we had talked about that as well. I'm trying to figure out where we were. This was the bacteriophage that we said is going to be a virus that actually infects bacteria and can actually be used in recombinant DNA technology to help insert into bacteria and other microbes genes that we want to change those microorganisms with. That's called recombinant DNA technology. So something in nature that we're just finding and using. So you can actually see some of these little pictures and whatever, but if I don't talk about it, we don't. Okay. Live animals, we said that in virology you have to either use animal tissues, bird embryos are great to use, and then just cell culture techniques. All of that being kind of expensive, aren't they? What'd you say, Jenna?
I said we should use Amen.
Just have chest pain. Okay, so but for the grace of God, they're sick. Somebody, you know. So there's our justice system is neither blind nor fair, and there are a lot of people. You know, there's some people that maybe shouldn't be around other people, but most people that we're calling inmates in our system should never serve again. Most. So just saying. So if somebody, I would really recommend if anybody gets the opportunity to actually go into a system and meet with some people and see what they're in there for, you might change your mind. You might. It really, really might change your mind about some stuff. Yep. So anyway, so looking at cytopathic effects, and we know what bacteriophages are. I was trying to think if I finished all of this. I don't know. I think so. So this actually brings up something that, again, maybe a little bit before we get to, but I want to get to this. Sometimes we can have viral infections that never really make us stop skipping and jumping. In other words, you know, we don't know. Just like I just introduced to you that you can have something as serious as hepatitis B and C, and you never even really know about that initial illness. It doesn't make you sick. It can make some people really sick initially. Actually, they're kind of the lucky ones, because if they live through that initial thing, they get treated, don't they? And some things you can just get cured for. But in other times, you don't really get sick. So there are a lot of viruses that we get exposed to that never make us sick. But if we get exposed to it, what will our immune systems produce? What kind of proteins? Antibodies. And are those antibodies going to be specifically against that particular antigen on that particular virus? Yes. And guess what? Sometimes the shape of that virus's antigenic outfit is so close to our own tissues that the antibodies that we make against those viruses end up attacking our own tissues. Guess what those are called? Those are called autoimmune diseases. Those are called autoimmune diseases. Type 1 diabetes is caused from that happening. That is a type of cross-reactivity. That's called cross-reactivity. So you end up getting a virus that didn't even make you sick, but it caused you to make an antibody that had such a particular shape that it could attack a particular cell of yours. Like the beta islet cells of your pancreas. The beta islet cells of your pancreas produce what? Insulin. So now you're making antibodies that are attacking your own beta islet cells. It's an autoimmune disease. And we think a lot of our autoimmune diseases, we know multiple sclerosis is also an autoimmune disease, isn't it? So we know that a lot of our autoimmune diseases, and we're thinking now even some other cases, is because of this kind of cross-reactivity. So something to think about, isn't it?
So it's making antibodies that attack your natural.
A particular type of your own cell. Yeah, so a particular type of your own cell. You know, I feel like, I know I can't draw, but I feel like if I draw this, it seems like everybody's going to get it. Right? Okay, yes.
So I saw polio on that list. Is that the one where they had like the iron lung and they go in the slug?
Yes. Yeah, so and I'm going to talk about polio later when we get to the respiratory system, but polio virus can affect the muscles that are responsible for you breathing. So it can paralyze those muscles. So Randolph Henry High School, back in the day, was the gymnasium. At one point, they brought in iron lungs because there was a polio outbreak in the county. And some people, there'd been enough people that needed to go into those lungs so that it was breathing for them to help them get through the acute phase of the disease. So this is something not in y'all don't even think about that, do you? You know why you don't? Because of immunizations. So anyway, guys, so here is an antigen that's on the outside of a pathogen, right? And it has a very particular shape, you know, because all species have their own identity, right? So this little antigen is repeated, this little shape, stop it and go away. This little shape, and it's invaded your body. So what did you tell me will happen if something that's foreign invades your body? You will make a what? You'll make an antibody. And the antibody that you make, I wish I had a different color. The antibody that you make will be a structure that has that wasn't the color, that has the absolute complementary shape to that. Do you see that? So this is the protein that you made. Oh, now it's blue. Okay, so this is the protein that you made, right? This is their antibody, and you made it in response to that antigen. So now your antibodies go around and gobble up anything that looks like that and takes them out. Don't antibodies attack, destroy, annihilate? But if this shape of this particular virus is so close to the self-antigens on your, like, say, beta islet cells of your pancreas, what would this antibody also interfere with? Those cells. And it would attack those cells. And then you start attacking those cells, and those cell numbers start to go down, and those cells produce insulin. So what's your insulin amounts doing? Going down. That's called the honeymoon phase of type 1 diabetes, where you've got, you know, if y'all know anybody who's ever been diagnosed with type 1 diabetes, they go through this period that's called a honeymoon phase, lasts about a year, where they're making a little insulin, but not a lot, and then eventually they just stop making insulin. Because what happened? They had antibodies that destroyed the beta islet cells that were cross-reacting. What? Huh? So that were how do I get rid of it? That were cross-reacting. Clear. Okay, what? All right. Yes.
Is there a way to get rid of the antibodies?
No, not really. You can do some, it would be an ongoing process if you tried to do that, because antibodies are in the body fluids. So blood plasma, interstitial fluids, lymph, blood, antibodies are in fluids of the body. You would have to do this continual type of thing where you are trying to get them out. Yes?
Are there any viruses that target antibodies?
No. So, no, antibodies are made to fight the viruses. But I see where you're going with that. It's not a bad question. So, at all. They're never bad questions. Okay. So these are some common virus families, general, whatever, but remember, if we haven't talked about them, then we won't include them yet on our list. I do want you to know there's something called viroids. Viroids are going to be even less complicated than viruses are, and they're just little particles, really, viroids are, that can be pathogenic to plants. So are we caring about anything that's happening to plants? Why? Because we eat plants, yes. So we do care about these, but I don't really talk about them in this class of infectious diseases for humans. But you all will have heard of them if you ever hear about viroids. So viroids are even a simpler particle than viruses are. Again, not considered living, but can attack plants.
So it's a particle that attacks plants.
Yeah, so it's like a sub thing of viruses.
A subvirus.
Yeah, and I'm not caring about them. But if y'all ever hear about them, you'll think, okay, I've heard about that before. And actually, we kind of do worry about them because we do rely on plants, don't we? We rely on plants, and we rely on meat that eats plants that we rely on. So we can be concerned about that as well. All right, so in chapter 6, I want to get through some of this in chapter 6 that is really going to be just some basic kind of definitions for you. So I'm going to roll along through this and hope that you all will get what you need out of this. When we talk about nutrition and what these microbes are going to need to actually survive, obviously they survive pretty well in humans, don't they? So they're getting what they need in you, or they wouldn't be making you sick. But when we're thinking about this, we can also think about it on a sort of a bigger level, macroscopic level, and think about these are sort of the same things that we need. So if you talk about an essential nutrient, it means something that they're going to have that they can't really, it's got to be provided by something else because they can't make it, right? So whenever, like in a nutrition class, you hear that something's an essential vitamin, an essential mineral, an essential nutrient, it means that the cell itself can't make it. It's got to get it from an outside source. It doesn't mean that all those other nutrients aren't, look at me when I say this, aren't quote unquote essential. They're essential too, but the cells can make them. Do you know what I mean? Okay, all right, great. And some categories are called macronutrients because you have to have them in such huge quantities. You all know that all living things are mostly made of what? Carbon, hydrogen, oxygen, and nitrogen. Y'all know that, right? And they make up those categories called carbohydrates, what? Lipids, what? Proteins, and nucleic acids. But then we also have things called micronutrients that, we don't need them in as big a quantities, and sometimes they're referred to as trace elements, but we do need them. You still need them for life. So these are just sort of some definitions for you there. You guys can, this is still, I wish I'd never touched that smart ink. Who knows how to get rid of this? Does anybody see? Am I missing something? Maybe just hit it. Maybe it just gets rid of it if you do that, right? Do you think? No.
Go back.
Okay, thank you.
And hit Microsoft Ink.
Where is that?
It's besides one. It's just a guess. I don't know.
So you think that's just going to? Yay! Okay. So when we talk about inorganic versus organic, again, organic matter is going to be those ones that have a ton of carbon. It's really carbon and hydrogen in certain combinations, but anyway, so there's organic nutrients. As we said, the big macromolecules, carbohydrates, proteins. So they've got lots of carbon, lots of hydrogen. And inorganic are things like water, H2O. You don't hear anything about carbon in that, do you? Salts, electrolytes, calcium, these types of things. So these are going to be the inorganic nutrients that we need. So you all hear about chemistry. You can take inorganic chemistry or organic chemistry, right? It is recommended that you take the inorganic chemistry first and then build on that with more complex types of chemistry studies with the organic chemistry. Anyway. And now there's some other, these are just basic definitions. Let the words help you with this. What does hetero mean?
Different.
Yeah, so it means different. So heterotroph is going to be an organism that has to get its carbon source, the carbon, the organic macronutrients from something else. An autotroph, so this is something different to eat. Troph is to eat. An autotroph is a self-feeder. So an autotroph, so CO2, the carbon from the atmosphere, can be used for these autotrophs. Phototrophs can use photosynthesis. They can use light energy. Photo means light. So they can use light energy as a source to take carbon from the atmosphere and change it into a huge molecule of sugar. That's a good trick, isn't it? We could stand outside all day long and we wouldn't be able to make sugar, would we? But autotrophs can do that. And a chemotroph are going to be those ones that have to get energy from other chemical compounds. So that's how we know that some of those organisms, especially microbes, that live deep where there's no light, like deep in the ocean where it's completely dark, or in these volcanic vents, they can take chemicals just from their environment and be able to change those chemicals around and be able to feed themselves, give themselves energy and nutrients to feed. So these are just some definitions. What'd you say? This one?
Yes, you don't want to purchase one.
Okay. And these PowerPoints are on, they're on Canvas. So don't think that you all won't be able to get to these. You will. So if you just write these words down, then you can kind of go back. This slide has them again. It has them on here. You see what I mean? It has them on here. I would want you to know under the heterotrophs, meaning that you have to get organic matter from something else. You can't self-feed. We said that saprobes, which we knew fungi are saprobes, aren't they? Saprobes are going to get their organic matter from dead matter. And we said that one of the wonderful functions of fungi, in case you ever, were feeling not nice about moles and stuff, but a great thing about them is that they're decomposers, and we need them in our environment. So they're decomposers. They can take dead matter, organic matter, and from the environment and recycle it. So that's what a saprobe is. And then we also talked about parasites, how some things can be parasitic, meaning that they're going to live off of something else at the expense of something else. Fair? So this is just a list from your book. And again, you've got more slides that tell you a little bit more about them, but if you have just the basic things that I said, it's going to be fine. If we put some prefixes in front of parasites, would these make sense? If something is an ectoparasite, it means it lives where? On the outside of your body. If it's an endoparasite, where does it live? It lives inside your body organ somewhere. And if it's intracellular, yes, exactly. So this is these are kind of obvious explanations for these. And an obligate parasite means that it can't live outside. Some of these other parasites can live for a bit on the whatever outside of cells, but an obligate parasite has to be inside of a cell. So these are just, again, some basic kinds of definitions that when we talk about things, we have, a lot of people get grossed out by this. I just was surprised that I was talking to a nurse practitioner, and the nurse practitioner didn't realize that we have mites on our skin. But you guys, we have mites on our skin, okay, that actually are just part of our microbiome. They're really, and I think I have one on your list of other things on like at the end of your list. I think I have like the Demodex. Do I have that?
Yeah, Demodex.
Yeah, so I have those, and the reason I say this is because some people in that microbiome, we said we need a balance, don't we? So we need everything being in balance because that actually keeps us healthy. But sometimes what are the most causes for problems in humans are oftentimes opportunistic infections from their own microbiome. True? So the reason, I think one of the reasons I put that Demodex and it's a mite, is that some people, if you've ever heard of rosacea, Have y'all ever heard of rosacea? Rosacea is an autoimmune disease. I have rosacea. There's some people with rosacea that have their immune systems get really jacked up against some of their own microbiome. And one of those are those mites that we all have on our skin. And we do all have them on our skin. We have protozoa in our bodies. We don't just have bacteria. I think people think that we just have microbiomes means it's just bacteria. We don't. We have all kinds of things living in and on us. And some of them are parasitic, but some of them really aren't. Some of them are actually mutualistic, meaning that they help us, right? That it's a mutual relationship. So when we think about nutrients, and I know that I've already talked to you guys because most of y'all have had me for AMP, so y'all understand what diffusion and osmosis is about. But when we're talking about nutrients, there has to be a way for any living cells, whether they are single cell microbes like bacteria or protozoa, or whether they are multi-celled organisms like fungi. Fungi can be either single cell or multi-cell, or helminths. They have to have a way to bring in nutrients. And so we talk about diffusion. Diffusion is a big way of moving solutes from one area of concentration to another, right? So most solutes move just without any energy down their concentration gradients. So they'll move from where there's high amounts of solutes to low with no energy required. So that's moving down their gradients. The movement of water, and we know water is just essential to all life, right? So the movement of water is referred to between two areas of concentration is called osmosis. So this is referred to as osmosis. And water will always move down its gradient. There's no way that you can move water against its gradient at a cellular level. Like we can do it if we have some pumps, and we can do it if we put in a ton of energy, right? But at a cellular level, which is where life is at a cellular level, water moves down its gradient. We can't move it against it, okay? So this is, do I have any questions about that? I think that's repeat for most of you all because you've had AMP and you understand how cellular processes work and how cells can get nutrients in and get rid of waste products and how cells keep their water balance, the inside and outside. I think most of you all, has everybody had that? So with osmosis. Okay. So when we talk about this movement of water, we do often talk about tonicity of a solution. And tonicity is just the idea of how that solution will affect cell size. That's what tonicity is referring to. So tonicity is, y'all can't see that at all, can you? I can't see it from here. So I know y'all can't see it. Can you? Okay, I'm sorry about that. But anyway, so tonicity is just the ability of a solution to affect the cell size. That's what when you're talking about tonicity. So if a solution is isotonic, it means that there's going to be equal amounts of water on the inside and outside of the cell. So is the cell size going to be affected? Water's just going to be moving kind of in and out, but it's not going to interrupt the normal cell size, right? Isotonic, an isotonic solution. If it means that iso means equal, so it means that that solution is going to keep that cell size just balanced. It's going to be balanced. They're going to be balanced between the outside water and the inside water is balanced. It's equal. Iso, equal. If a solution is hypertonic, what does hyper mean? A lot. It means it has a lot of solutes. If it has a lot of solutes, it means it doesn't have very much what? It doesn't have very much water. So if a solution is hypertonic, then the water inside of cells will move out. And what will that affect the cell size? What will the cell size do? Shrink. And can cells some cells can't even survive that. Some cells can, but they're not going to function as well, are they? And if a solution is hypotonic, what does that mean? It means it's less solutes, more water, and you put a cell in it, and there's more water outside the cell than inside the cell, what's the water going to do? Move into the cell, and the cell is going to do what? It could burst. It could expand, and it could burst. It could. And a cell that bursts is called lysing, right? That's lysing of a cell. So it could affect it. So we know that cells have to drink and eat, essentially. Don't they? Don't they have to be able to drink and eat, and they have to be able to get rid of waste products? And the way that they do that is through, if it's a solid, it's through distribution. If it's water, it's through osmosis. So great. These processes, endocytosis, the process of a cell bringing things in. Phagocytosis is the process of a cell eating, right? Pinocytosis is just ingesting of liquids or oils, bringing in. So it's sort of the same thing as endo. I don't ever ask you about pinocytosis, but okay. And do temperatures have to be just right, guys? Should temperatures be just right? Yeah, I'm mixing up the slides this time. And I'm not going to ask you these terms. I'm telling you now, you can look at this, but I'm never going to ask you these questions. But what we know for most organisms, we know we're at optimal temperatures that they need to survive. So if we're looking at Celsius, and you all understand that Celsius, 37 degrees Celsius is like 98 point whatever for Fahrenheit, right? So I think most people know the difference in Celsius and Fahrenheit. So if you're looking at some of these organisms, or you're looking at some of these temperatures and optimal growth, then certainly if we had pathogens that we were worried about getting from a human, now we know if they're causing problems in humans, what temperature do they like? Okay, 37 degrees, right? But some of them we know can stand higher, and some of them we know can stand lower. So we definitely want to give them what they need to live, but we can use temperature to help us isolate. Today, y'all are going to see your plates, and I thought y'all did a great job with the plates, seriously. But y'all are going to see your plates today, and you're going to actually see in that first area that you put things in, it's just solid growth. But where you all sterilized your loop and you went out to the second quadrant, third, you're going to see isolated colonies. And we can only start our identification process of pathogens if we have isolated colonies. Because if I took a mixture of something and did biochemical testing, would I be able to trust any of the biochemical testing? No, not at all. All the tests would be positive, but that's because we had a mixture. We didn't know what was doing what, right? You have to have pure cultures and isolated colonies. So we can use temperature for this way, but I'm not going to ask you the names of those. Same thing with gases. Do we know that there's some bacteria that cannot survive in the presence of oxygen? What do we call them? We call them anaerobes. If we had an organism that had to have oxygen, an aerobe, it's an aerobic organism. I'm not going to put it in an anaerobic environment. I wouldn't get it out, right? But we know some microbes, and I'm going to introduce one to you right now. We know some microbes that have to have a microaerophilic environment. What? Have you heard this one before? Microaerophilic? A microaerophilic organism can only survive and grow in reduced oxygen environment. That it needs to have some, but it has to be very reduced. I'm going to give you an example of a microaerophilic organism. You ready? It is going to be Campylobacter Jejuni. Campylobacter Jejuni, it's a bacterium. And it causes, it's caused by food poisoning. So usually somebody's ingested it through contaminated meat or foods. Has anybody ever heard of that? Yes. All right, so if you go to your list and you see it, Campylobacter Jejuni, I'm checking it off. Okay, thank you. Thank you, Sarah. So you're checking it off. You'll see that it's a bacterium, and it causes a pretty serious gastroenteritis. It is from contaminated food sources. If I were to go right now up a mile down the road to a food line and take swabs down their meat counters, we could grow it. But to grow it in our lab, what must we do?
Reduce the oxygen.
Yes, we're going to have to reduce that, and then we would get it out. Now, this is a fairly common, probably, honestly, I'm going to say, well, maybe not y'all, I'm sure I have though, most of us have had this. Most of us have been sick with this. We were sick for several days. We were really sick. We were making bargains with supreme beings, just if we could live another day, you know. So we were pretty sick, but we got over it, right? Some people get so sick though that they end up getting cultures done, and then they get proven that they have it, you know, because the cultures come back, stool cultures come back positive. One of the reasons I really want you all to know about it is not because it's so common, which it is. It's very common. But it's also because it has been linked to something that happens rarely, but it does happen. It's called Guillain-Barre Syndrome. Is that that's on your notes, right? Has anybody ever heard of, and the way that people pronounce it that have the accent is Guillain-Barre, whatever, Guillain-Barre Syndrome. So does anybody ever heard of that syndrome? Yes. Have you been?
Yeah, that's what it was asking. Blood, you had that childhood disease, it's all
Yes. Okay. So this syndrome is really being linked to having a prior history of having Campylobacter infection, and Campylobacter is pretty common. So it's pretty rare that somebody would have this, but this syndrome is serious because you get a progressive paralysis. And it can happen at any age. Little kids, this will happen, where they'll start becoming progressively paralyzed, and it usually progresses from the extremities all the way and can go all the way to affect respiratory function. The good news, if you're looking for good news, the good news about this syndrome is that it typically will start to go away after a time, and you'll get the feelings back and be able to move again. It is not affecting sensory. It is only affecting motor. So people are becoming paralyzed, but they can still feel every time you give them an injection. So don't think they can't. They can still feel everything. They just can't move. It can take up to a year to get back feeling, to get back movement, for the paralysis to go away. And on rare occasions, some people don't get it back completely. Like they'll get a lot better, but they might not get it back completely. Yes?
Can you say it again?
Guillain-Barre. And as I said, sometimes they call it Guillain-Barre. Do you see it in your notes? It's Campylobacter Jejuni. It's bacteria. Campylobacter.
First page on.
First page, bottom something.
Back of the first page.
Back of the first page, bottom something.
Yeah,
Oh.
stuff.
You thought I was talking about what? What do you think I was talking about?
I don't know, you said it like causes something, so I was looking for what you were saying.
Oh, it causes like a gastroenteritis. So it causes diarrhea and vomiting. Diarrhea and vomiting. And you know, and most of us that have had it have never gone and got diagnosed we had it. It's just that it's rare. This is one of those things that we do think can follow that infection. And what it is your immune system doing that. Your immune system is cross-reacting, attacking those motor neurons. Do you get what I mean? So, you know, guys, what we need to realize is that a lot of times the reason people are so sick is because, you know, we may get something like a virus or whatnot, but it's our immune response that's making us really sick, because our immune response is bringing mucus to the site, you know, and it's raising our temperature. And so our signs and symptoms are coming from our immune response that's attacking. Do you get what I mean? And so this is one of those things. And so I do want you to know about Campylobacter Jejuni. It is a very common cause of food poisoning types of gastroenteritis on rare occasions. Does anybody know anybody who has Guillain-Barre? Anybody? You work in any hospital any bigger than maybe the little local ones, and you'll have patients in your ICUs that are with that syndrome. You'll have patients. And the good news about those patients, because it's frightening, you can imagine how terrifying that is that you're just saying, look, we just gotta keep you stable, and you're gonna get back. You're gonna get back. You're not gonna be paralyzed forever. But it can take up to a year. I remember a little 6-year-old that had complete paralysis, even respiratory. And the worry is that when somebody's completely paralyzed, can they get something else? So that's the worry. So that's why they're in the ICU. And that's why it's the worry. But then they start getting it back. Yes.
How high is the mortality rate?
You know, I have not ever really looked it up. It's a really good question. You could we can easily do that. Look, he's got his phone. And I'm going to say, okay, look it up. Go ahead. We don't usually have phones out in here, but do look it up. I'd like to know. What's the mortality rate of Guillain-Barre? So then there's some microbes that can only grow if we have enough CO2 in the atmosphere. So there's got to be, some of them like a higher amount of CO2, and so we've really got to get that increased CO2. Neisseria is one of those that really likes, it's Campylobacter, it loves CO2, and it's going to need that carbon so that it can make its own organic products. So Neisseria, if you suspect somebody had gonorrhea, you just going to leave those plates out just on the counter? No, you're going to increase the CO2 and give it to them. It's food to them. So whatever. You don't have to know that's one of them. I just want you to know these are some of the things. So we think about temperatures. We think about environments. We realize that we think about pH. We think about these things that they're going to need and where they're going to grow the best. And these are just considerations when we're thinking about them. Symbiosis means what? Symbiosis means to what? Partnership. To live in harmony, right? So partnership, if you will. That's fine to say it that way. So when we think about symbiosis, I wish I could actually read up close. When we think about symbiosis, this is, yeah. So there's different types of it. Mutualism is sort of like what you think of the word mutual. They're both kind of mutually benefiting. Commensalism. This is, again, I sort of think about this as sometimes we hear about our microbiomes as being commensalism, but they're really not. It's more of a mutualism. But this is one partner is neither harm nor benefit. So the partner's receiving the benefits, but the other one is not really harmed. Parasitism, that means that one of them is going to be paying a price. Isn't that right? For the other one surviving. So these are just three types of symbiosis, but they're symbiosis, right? A perfect relationship, even if you're a parasite. If you're a parasite and you're benefiting off of a host and you're causing a little bit of harm, would you want to destroy your host? No. So a perfect parasite evolves to not destroy the host. And it's the ones that have become these perfect parasites that end up being spread through populations so much because they're not killing someone. Like, let me give you an example. Ebola, and if I hadn't talked about it, you can check that off your list. Ebola is a virus. So that's a virus. Anyways, going back to, okay. All right. I'm having fun watching all of these colored pages. I haven't done these colored pages before. I've never given people these. So I have given the list at the end for people to study, but I've never done this. So I'm kind of like these colored pages. Ebola. Ebola is a virus. It is an obligate parasite, isn't it? And you know, you might, when you just think about Ebola, you might think, wow, that's a perfect parasite because it kills about 90+% of the people it infects. But is it? Is it a perfect parasite? It really kind of isn't, because it kills so quickly and so rapidly that we're able to quarantine people down, right? And so it is not jumping to and staying, which we actually kind of glad of, aren't we? So, but anyway, so these are just terms that are used for relationships. Antagonism means what? Yeah, so working a species, well, what do we want to say from here? I don't know. So antagonism means working against, but antibiosis means producing things that would really hurt something else. So penicillium is a mold that produces penicillin that we know is an antimicrobial, isn't it? It works against some bacteria. It actually destroys some bacteria. So we understand that. Synergies. What'd you say? Antagonism, so working against, so making something that you would make. Antibiosis. So when you think of bios means life, right? So against other living organisms. So making a product that could be used against other living organisms. And then we talked about synergy, which is the same. And we've talked about biofilms, haven't we? We said biofilms are really like these perfect communities, and they have mixed species in these communities that are forming. And are they talking to each other? And how are they talking to each other? Through quorum sensing, which we know. And if we can figure out how they talk to each other, would that be a method that we could maybe use to interfere with their communication? Yes. So that was the importance of that. And I had already introduced you all to that. So great. Do we know how bacteria grow? Prokaryotic or binary fission. That's what it's called, isn't it? It's a very simple kind of process. What we know about these microbes that you all have on your list that you certainly don't have to know for this introductory class, but what we know for every one of those microbes that you have on your list, which are the more common pathogens, we know exactly what kind of doubling times they have, which are referred to as generation times. So how quickly they can divide, how quickly they divide. So I would just want to give you some examples. You certainly don't have to know this, but there's some that can divide every 10 to 12 minutes. There's some that are going to take a little bit longer as far as minutes, and then there's some that can take days. So is this going to affect our results time from a micro lab, getting the results back? It potentially can. So what would we want to do instead? Instead of waiting for something that's going to take days and weeks, we want to do a what? But I didn't hear you. Come on. What kind of test can we do? Because by the time that you've taken weeks and months, you've already built a what? So you can do some indirect testing instead, right? Right? So that's exactly right. I mean, we can still culture it, but it's just going to take a long time for some of these. Most of the pathogens that we know, and this is the last thing, and I know I got to leave because there's a class coming, and we're gonna walk down to the lab, and all I want you to do in lab today is look at your plates and look at some things about your plates. But before we look at those plates, I need to give you some safety guidelines, okay? So anyway, but here's what I do want to talk to you about. Does it make sense to you all that when a microbe first enters, and this is a test tube, but you can think about it as your body, but when a microbe first gets introduced into your body, there's going to be this little period of time that it's acclimating to your cells. This is actually referred to as a lag phase. Then all of a sudden, there will be exponential growth, which means that the doubling time, I mean, it's really growing rapidly. Then there will be a stationary and then death phase. What I want you to get from this is this. Where microbes, microorganisms that cause disease, where we can most effectively treat them with chemotherapeutic agents is when they're in this phase. They're most responsive to drugs when they're in this phase. So do we need to catch it here for most diseases? This is where we would want to catch it, is when they are in this phase. If you've got somebody that's already, it has been in it too long, it's going to be more difficult to hit them and to treat them. So that's kind of what I wanted you to get from that slide. And I highlighted it for you so that you could kind of see on this that when they're in that exponential phase is when they're the most pain. Now, can you be spreading disease even early on? Yes. 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 All right, so do y'all have any notes?
Yes. Can you ask her if we have the blood tubes?
Oh, to see your plates?
Yeah.
Yeah. Is that okay? Do you have something going on in here? Is that all right with you to see it? What'd you say? It's not okay with you?
Yeah, it's okay.
Oh, okay.
I'm saying the assignments that she gives us, basically we already do it at home.
So it's okay if you stay with me for 10 minutes. I'll ask her. I'm gonna ask her right here.
Do we get a